Well, the hope is a lot of times when we're doing a molecular-based approach that we're trying to go after events that we think occurred very, very early in the cancer development process and trying to actually eradicate all of the cells. So the concept is it's much more of an attempt to get a curative attempt. The trick is today we don't always have drugs that are targeting events that occurred really, really early in the process. The ones that we've had for the most part have been things that are occurring later in the process. So we often are removing big subsets of the cells, but we're not able to get all of them today because we don't always have agents that have occurred really early. The best example in other cancers would be the BCRA-ABL inhibitors in chronic myelogenous leukemia, which actually hits the very first event that occurs in those tumors. In myeloma, the similar type of events we just haven't developed drugs, but there's active development trying to get drugs to those targets. Some of the most advanced things that we have for doing molecular-based treatments, the one that we've been working on for probably the longest time has been looking at patients who have activating mutations of FGFR3. This occurs in about 4% of all myeloma patients in what has historically been a high-risk subset of patients that have the 414 translocation. And what we're now starting to see is those agents have matured. There's really good data presented, not at this meeting, but the last Myeloma 2019 meeting, really showing for the first time one of those agents working in a patient where some of the cells had the mutation and some of them didn't. And in that example, it completely eradicated all the cells with the mutation, unfortunately leaving the tumor cells that didn't have the mutation, but it was really good proof that if you had a patient where that mutation was one of the earliest events, we could have potentially eradicated all the tumor cells.